Hi and welcome back to another A-level biology video. This one's all about mitosis. We'll be looking at the 4 stages of mitosis and learning how to identify them under the microscope. Let's get started. In interphase, the cells look like this. The 46 chromosomes inside the nucleus exist as chromatin. Chromatin is that indistinct mass of unravelled DNA. We cannot see the individual chromosomes under a microscope during interphase. During the synthesis phase of interphase, each chromosome replicates, making 2 sister chromatids. The sister chromatids are held together by a centromere. After the original chromosome has been replicated, we still call the whole thing one chromosome. The process of semi-conservative DNA replication is covered in another video. When the chromosomes have been replicated and the cell is ready to divide, mitosis begins. Mitosis occurs in 4 stages. The first stage is prophase. During prophase, the chromosomes condense and the nuclear membrane disintegrates. The chromosomes condense because the DNA coils up extremely tightly around histone proteins. The chromosomes can now be seen under the light microscope. Remember, we have 2 sister chromatids making up each chromosome. The second stage is metaphase. During metaphase, the chromosomes line up at the equator of the cell. Their centromeres become attached to the spindle fibres. The spindle fibres are made by small organelles called centrioles, which sit at opposite poles of the cell. The third stage is anaphase. During anaphase, the spindle fibres contract, separating sister chromatids and dragging them to opposite poles of the cell. The chromatids appear V-shaped when they are seen under a microscope. The fourth and final stage of mitosis is telophase. During telophase, the chromatids reach the poles of the cell. They are now referred to as chromosomes again. The nuclear membranes form around them and the chromosomes unravel. Finally, the cytoplasm divides in 2. This is called cytokinesis. Mitosis produces 2 genetically identical daughter cells. This process is occurring constantly in living organisms, allowing them to grow and to repair damaged tissues. Here are some images of cells under the microscope. See if you can pick out any cells that look like they're in interphase, prophase, metaphase, anaphase, and telophase. You can pause the video if you like. These cells, circled red, are in interphase. We can tell this because the genetic material is contained in the nucleus, but we can't distinguish individual chromosomes. This cell is in prophase. The nuclear membrane has disintegrated, and the chromosomes are now visible because they're condensed. This cell is in metaphase. The chromosomes are fully condensed and they're lined up more or less on the equator of the cell. These cells are in Anaphase. We can just about make out the V shape as they are pulled to opposite poles of the cell. Finally, this cell is in Telophase. We can tell this because the chromosomes have reached the opposite poles of the cell. There's a good gap between the two sets of DNA, but the cell hasn't split in two yet. Cytokinesis hasn't been completed. So that's it for another video. As always, there will be resources linked down below in the description box. Please like and subscribe, and I'll see you in another video. Thanks for watching.